falling under Task 3.2 on the development and adoption of simple, rapid, and inexpensive ICT-based tools for detection and quantification of plant pests and pathogens, STMicroelectronics has been developing a full molecular analysis workflow on a qPCR portable device called Q3 to test model crops that may be contaminated with mycotoxin produces fungi. Research in the field is crucial in attaining food security because mycotoxins are increasingly acknowledged as significant contributors to foodborne illnesses, especially in sub-Saharan Africa. But what are mycotoxins?
Mycotoxins are harmful secondary metabolites produced by filamentous fungi, also known as molds, that by growing on cereals, fresh and dried fruits and vegetables may compromise grain quality, crop yield, and ultimately cause severe mycotoxicosis in humans and livestock. Among the most harmful mycotoxins, aflatoxins produced by some Aspergillus species and fumonisins that are produced by some Fusarium species raise particular concern due to their association with liver and esophageal cancer. But many other mycotoxins exist in nature that have negative impacts on our health status and well-being. It is estimated that 1/3 of agricultural commodities produced worldwide are spoiled or wasted during the post-harvest stages, with increasing number admitted for some regions and crops. Recognizing the relevance of mycotoxins and the issues these compounds represent for food security in sub-Saharan Africa, the EWABELT Project is developing an innovative approach spanning from improved storage technologies and the seizure support system to early detection of molds development in the store commodities. This is exactly what STMicroelectronics is doing within the consortium with the development of the portable qPCR tool. The University of Sassari, along with STMicroelectronics, have selected 8 mycotoxin-related genes that are considered as specific markers of the ability of fungi to produce major mycotoxins occurring in cereals. Such genes are detected with the portable qPCR tools, which represent an alternative mitigation strategy to cope with the harm posed by these toxin contaminants to food security worldwide. But let's see how it works.
qPCR is the gold standard molecular technique allowing for specific DNA or RNA sequences in a sample to be analyzed. If such sequences are present, they will be exponentially replicated to billions of copies, so that they can be easily detected, typically through fluorescence measurement. Although qPCR guarantees the best possible sensitivity and specificity, it also has some cons which have limited its diffusion so far. Mainly, it is quite complex and it requires bulky and expensive instrumentation. Tackling these limitations is the objective of STMicroelectronics within the EWABELT Project, with the development of an innovative molecular analysis workflow, which includes 2 steps. First, the extraction of DNA from the crop sample. We have set up a simple protocol for maize flour and peanut specimens, which requires very little equipment. This protocol was tested on samples provided by the University of Sassari. Then, in the second step, the extracted DNA is analyzed by qPCR on a system developed by STMicroelectronics, which is called Q3. This system is composed of 3 elements: a single-use cartridge, a portable instrument, and a software application. Besides this, specific reagents for each target DNA sequence are needed. The cartridge is the core of the system where the reactions occur. Since qPCR requires precise heating and cooling cycles, the cartridge is built around a silicon chip which integrates a miniaturized heater and temperature sensor. The chip is produced according to microelectronic standards that guarantee a high quality level. The Q3 instrument is far smaller than traditional qPCR equipment. It drives the chip and features a compact fluorescence microscope to measure the optical signals coming from the reactions. A dedicated software application guides the user through step-by-step instructions. At the end of a test, the output files can be uploaded to the PlantHead platform developed by Occam, whose demo you saw before. Also, very importantly, the results are displayed in a very straightforward way, that is, sample positive or negative for each of the analyzed mycotoxin-related genes. So, thanks to the sensitivity of qPCR, we can perform an early detection of mycotoxin-producing fungi, which can promptly trigger containment actions to try and save the crops, or at least limit their waste. So, we have a series of advantages compared to traditional qPCR equipment: portability, lower cost, and ease of use, without sacrificing performance. All of these allows taking molecular analysis out of the big, highly equipped laboratories and making them more accessible. And this is exactly what the University of Nairobi is experimenting with.
Abigael Ouko. I work at the University of Nairobi, Kenya. I am a geneticist. The University of Nairobi was chosen to be the first end user to be trained by ST on all the molecular analysis work. The first working session was organized during the WP3 capacity building workshop in Kenya in October 2022. A group of researchers and students were trained on the developed DNA extraction and qPCR procedures. After a few hours of training, they were already able to successfully run the molecular tests. Since then, numerous relevant tests have been conducted at our mycotoxin laboratory. Peanut seeds without skin and peanut seeds with skin were used for the experiment. And peanut seeds without skin gave best results. Indeed, when peanut skin is removed, it is easier to visually distinguish the magnetic beads used for DNA extraction from the peanut debris. Removal of the peanut skin also reduces obstruction of pipette tips during DNA extraction. We therefore modified the protocol to always use peanut seeds without skin. Then, using the Q3 qPCR machine, we have been able to detect peanut control DNA, which indicates that DNA extraction has been successfully performed. Some mycotoxin-related genes with clear indication of the positive or negative calls for each analyzed mycotoxin, and these are always shown below the qPCR graph. The plots in our experiments showed clear curves of the peanut DNA controls. In some cases, the curves had an early rise, indicating high DNA concentrations, unlike in some experiments where the curves had a slightly later rise, indicating lower DNA concentrations. Reports generated after each experiment illustrates detailed but easy to understand results from all the 4 analysis wells. In doing so, it enables one to tell whether pipetting in each of the wells was precise or not. Although further testing is required, molecular analysis with the Q3 device can represent a valuable and easy to use tool for further accessibility of scientific instruments at all levels, enabling cost reduction of sophisticated molecular tests, sensitivity in detection of mycotoxin producing fungi, simultaneous monitoring of many different mycotoxin producing pathogens, and evaluation of the potential risk for foodstuffs. Combined with traditional and other innovative approaches like the decision support system, also known as the DSS, for real-time monitoring in silos and storage facilities under development by Cranfield University, these instruments will allow for toxic fungi detection to eventually enrich the existing literature and provide solutions for healthy food for all.